Hey, so welcome to our lesson 10.2. Today we're going to be covering meiosis. Now I emphasize the me portion of it, but really it's meiosis. That just sounds really similar to mitosis. So I just emphasize the me in meiosis. So we're in unit 10.2. We're taking a look at meiosis. Oops, excuse me. That's why I emphasize the me portion of it so we can spell it correctly. Meiosis. All right, there we go. So starting off, some difference in terminology. Diploid versus haploid. All right, so where we left off. So again, as Mendel theorized, eukaryotic organisms have pairs of every gene. Again, Mendel worked with his P plants, was able to figure that out. But again, he did that without knowing about chromosomes, without knowing about DNA, without knowing about genes. He just figured organisms have two of each trait, two copies of each trait. So as we know today, though, so today we know that those matching genes are on chromosomes, so these long stretches of DNA, and the fact that there's a pair of them, they're called homologous chromosomes. But again, Mendel didn't know any of this. This is stuff that you know that one of the most famous scientists in the world didn't know. So you're already ahead of him in your education. Here is our first term then. Diploid. So cells that have those matching pairs of chromosomes, so homologous chromosomes, they're said to be diploid. So di is a pretty common prefix in science. It generally means two. So two ploids, two ploidy. The symbol for diploid is 2N. And what that symbol stands for, N represents so the variable N represents the number of chromosomes a species has. And the constant 2 kind of goes along with diploid. 2, you have 2 of each of those unique chromosomes. So as an example, I think we're all pretty well aware of humans. Our N number is 23. We have 23 unique chromosomes. So diploid is 2 times 23 for a total of 46 chromosomes. That's total. You have a pair of 23 individual chromosomes, 46 total chromosomes. Again, why do you have a pair? Well, one from mom, one from dad. Kind of continuing on with definitions here. Why do we have so many different names for the same thing? I'm not exactly sure, but we also call our diploid cells somatic cells. Hopefully that term sounds somewhat familiar. We have used it a few times this year, but if not, don't worry about it. Now is when you need to know it. So our diploid cells, the ones that have a pair of all of our chromosomes, are also known as our somatic cells. And most of the cells in your body are somatic cells. Your skin cells, your brain cells, your liver cells, your stomach cells, all somatic cells. They're all diploid. They have a pair of each chromosome. But, in fact, I'm going to move this out a little bit. I want it to be a main point. So, out here, but. But during reproduction, only one gene gets passed on to your offspring. And that's Mendel's law of segregation. You might have a pair of each gene, but you're only going to pass one gene on. And that's kind of bringing us to what we're going to look at today. So I'll change that, sorry, from gene, although we could use the word gene. I'm going to keep it at chromosome. So cells that only contain one of each chromosome, which generally might be the gene as well. Genes are found on chromosomes. Haploid. There's our other term from this section, diploid and haploid. So our cells that only contain one of each chromosome are called haploid, and they're symbolized 1n. Almost sounds like half. So whatever your n number is, you only multiply it by 1. So human haploid cells are only 1n. Again, just because we like to be confusing and throw a ton of words out there, haploid cells are also called gametes. Gametes. And one more time, just because, well, we like using a lot of words, even though few may do. Gametes in males are called sperm. In females, they're called eggs. All right. So gametes include, in males, we know them as sperm, and females, eggs. For seed-bearing species, we don't normally think of plants as reproducing, but they certainly do. So in seed-bearing species, like plants, the male gamete is called pollen, and the female gamete are called ovum. So plants have slightly different gametes, so they really function in much the same way as sperm and eggs. So all of these are gametes. Now we're on to kind of what we are covering today. Okay, there it is, title of the lesson. Gametes are formed in a process we call meiosis. Again, I'll oftentimes say meiosis just to emphasize the ME and make it sound a lot different than mitosis, because meiosis and mitosis sound similar, whereas meiosis and mitosis at least has a little bit of a difference. But when you continue on in biology and you have a professor someday and you go to talk to him, don't call it meiosis. That's just the way your high school teacher pronounced it. It's really meiosis, which just, again, sounds unfortunately a lot like mitosis. So let's take a look at the process of meiosis. So first thing to note, meiosis and mitosis are very, very similar, for better or worse. So if you guys remember mitosis from a couple of weeks ago, this process is going to be really similar. You're going to notice a lot of the same things, a lot of the same structures, a lot of the same vocab, and I'll really try to emphasize where the differences are between meiosis and mitosis. So starting off, big difference. Big difference right off the bat. Meiosis occurs in 2 stages. So let's jump right in. They are collectively known as meiosis 1 and meiosis 2. So starting off with meiosis 1. So this should look familiar to you. Now, I should point out I'm going to use 4 different colors, but what you'll notice about these 4 different colors is that 2 of them are very similar. I've got red and orange, very similar. They're going to represent a homologous pair of chromosomes, like one from your mom, one from your dad. And then I'm going to use light blue and dark blue. Different colors, but very similar. One from mom, one from dad. They're homologous chromosomes. Start by drawing these guys in our dissolving nucleus. Again, for right now, it doesn't particularly matter where. They've only just condensed. They've only just gone from that spaghetti chromatin shape to that more condensed chromatid shape, the X-looking shape. And this all happens in, hey, this should look really familiar to you, prophase. Got the 1 after it because, well, this is the first time that it happens. It's prophase 1. Spindles form. So we have that occurring, very similar, almost identical. In fact, it is pretty much identical to mitosis. The chromosomes condense. Into chromatid. So there's our D telling us that they're now visible. They don't look like spaghetti anymore. They're condensed. They can see them. The nucleus and the nucleolus are going to dissolve. We don't really want them. They just kind of get in the way for the time being. So they're going to dissolve during prophase 1. And then here is the new thing. Here is the new term, and this is what happens in prophase 1 of meiosis 1, but does not happen in prophase of mitosis. So this is a big difference here. If you want to put a little star next to it, homologous chromosomes form what is called a tetrad. I'm going to kind of blow this up. Again, like I said, the two similar colors, in this case red and orange, are going to represent homologous chromosomes. And they're actually going to kind of join together and overlap a little bit. It's like they're holding on to each other right there and right there. We'll talk a little bit more about that next week. But this right here is a tetrad. Tetrad, it's 4. Tetra is 4. So 4 chromatids. 2 orange, 2 red. Again, we've been through S phase, we've duplicated our DNA, so we've got 4 total sister chromatids that are all kind of joined together there in what we call the tetrad. Draw our drawing here. There's our tetrad. And here's our blue tetrad. So following prophase 1, you guys may have guessed, we have metaphase 1. Again, all that work you did back when we did mitosis is paying off for you now. Prophase followed by metaphase. And again, here is the big difference. Put a star next to it if you'd like. It's the tetrads, not the individual chromosomes. It's the tetrads this time. The tetrads are going to line up along the metaphase plate. Again, this kind of imaginary line down the middle of the cell is the metaphase plate. And we've got those tetrads lining up. Kind of out of order here. Probably should have had this occur first because it's actually the spindle fibers that are pulling those tetrads around. But we're going to have the spindle fibers attached to the chromosome, the entire X this time. If you remember back to mitosis, they attach to sister chromatids. This time, they're going to attach to the entire chromosome. As you can imagine, in our next step here, try to get the right colors going in the right directions. We have entire chromosomes separating. Again, remember back to mitosis, the X is split here. We pulled sister chromatids apart. Well, not pulling sister chromatids apart. Now we're just pulling the entire chromosomes in opposite directions. Again, hopefully you guys have guessed. Following metaphase, following metaphase is anaphase 1. In a little star, this is a little bit different. The homologous chromosome, not the sister chromatids, but the homologous chromosomes get pulled to opposite poles of the cell. So again, we're doing basically the same steps as mitosis, but a little bit different. We're separating sister chromosomes as opposed to sister chromatids. We're separating those homologous pairs. We'll go ahead and flip over to the next page here. You could probably guess our next step here. Move the camera up. We're now in telophase 1. We're also going to combine that here with cytokinesis 1. They're kind of occurring at the same time, just like they were in mitosis. So telophase 1, cytokinesis 1. And very similar to mitosis, we're going to have the cleavage furrow, divide the cells, in some species, the nucleus and nucleolus reform. The DNA decondenses back into chromatin. So sometimes this happens, sometimes it doesn't happen. We're actually not going to show this in our diagram. We're going to keep our cells nucleus free and keep our chromosomes in chromatid form. But just so you're aware, sometimes it does happen. This part is important. In no case, never do we continue to duplicate the DNA. We're not going to do that anymore. We don't need to. We're set. We've got what we need. S phase is not happening for these cells ever again. It's at this point, so using those terms we started the day with, it's at this point, at the end of telophase 1, that our cells are now considered haploid. We're not done yet. We still have all of meiosis 2 to cover, but it's at this point that they're now considered 1N because well, not considered haploid, excuse me, because they now only have one of each chromosome. They no longer have a pair. So our reddish colored ones, we've only got one chromosome each. And our bluish colored chromosome, only one in each cell. We no longer have a homologous pair. They've been separated. All right, and so begins meiosis 2. And hopefully, as you guessed, meiosis 2 is going to start with a very familiar phase at this point. Prophase 2. We got really creative with the naming, I know. Hey, it makes it easy, though. So during prophase 2, it's going to look very similar to prophase 1 in mitosis. The mitotic spindle is going to reform. So we've got these guys reforming. We're going to enter metaphase 2, where, as you saw, the spindle fibers are going to connect to the sister chromatids. So we no longer have homologous pairs. We don't have to worry about that. So the spindle fibers are going to attach to the sister chromatids, and they're going to pull those chromosomes into line on the metaphase plate. And as you might have guessed, I'm going to flip the page here, but we're going to move now into anaphase 2. Wrong color. And in anaphase 2, this is where we're going to separate the X's. So now we have the sister chromatids separating. Separating, not separating. I hope so, especially now that I've pointed it out to you. And I imagine we're getting pretty used to this at this point. Anaphase 2 is going to be followed by telophase and cytokinesis 2. So I'm kind of showing the DNA starting to unravel now. It's not the tight chromosome that it was up here. They're starting to unravel. Our nucleus is reforming. Our spindle fibers are breaking down. Our cells are cleaving. So we have our cells cleave. Our nucleus and nucleolus reform. The DNA decondenses, so it's going to unravel. It's going to go back to the chromatin spaghetti-like form. And our results are going to be 4 not genetically identical gametes. So if you guys recall back to mitosis, we always made a big deal of saying we're going to end up with 2 cells genetically identical. There were a lot of questions on that in your guys' homework on the labs. Mitosis always results in 2 genetically identical cells. Well, meiosis we're going to end up with genetically unique cells. Now, not entirely unique, I suppose. These 2 are going to be the same. We're going to have some blue. And again, we're back to that spaghetti-like chromatin form. We're going to have some orange mixed in there. The DNA has unraveled, but they're going to be different from this one, which is going to be some red and some dark blue. So these are not genetically the same. These are haploid cells, and they're different. Again, maybe you got, maybe these gametes have mom's beautiful blue eyes, and these gametes have perhaps dad's beautiful blue eyes. Just kind of a difference here. We'll see which one you pass on to your offspring. But there we go. These are our gametes. Those are the cells you would reproduce with. Genetically unique cells. And so that's how you go from 2N down to 1N. Hopefully this was helpful. Looking at meiosis, how it differs from mitosis. And again, hopefully it allows you to see, hey, that's how we go from a diploid cell down to a haploid cell. Feel free to let me know if you don't think it was all that helpful. We'll try again. If it wasn't so helpful, we'll try again. See if we can make it more helpful next time. Your input is always enjoyed. Well, have a great weekend. I'll see you guys next week.